Emily.
And I'm Cameron, and today we'll be explaining enzyme specificity.
Before we get started, let us take a moment to explain what enzymes are made of. Enzymes are proteins that are composed of long chains of amino acids known as peptides.
When a specific enzyme is formed, it is made by stringing together between 100 and 1,000 amino acids in a unique and specific order. This amino acid chain then folds into a particular shape, allowing the enzyme to catalyze one specific reaction, or in some cases, a small group of closely related reactions.
Enzymes act as biological catalysts by lowering the activation energy, ultimately speeding up the reaction that occurs within cells. Before we explain how enzymes speed up a reaction, let's give you a better idea of the anatomy of an enzyme and the components that go into the catalyst process.
Enzymes affect specific compounds, also known as substrates. There are special regions on each enzyme called active sites where these reactions take place. These active sites are regions on the surface of the enzyme that are formed by the way the protein folds up on itself in its tertiary structure, also known as its three-dimensional shape.
Only substrates with the correct shape can fit into these active sites. However, a substrate's ability to fit into the active site is not enough for catalysts to happen. These molecules also need attractive forces to aid in keeping them in place on the enzyme. The attractive forces involved in the active site can be hydrogen bonding, permanent dipole-dipole forces, instantaneous dipole-induced dipole forces, also known as London forces, or electrostatic attraction between oppositely charged groups.
The binding process is so selective that the enzyme's active site discriminates between two enantiomers, or mirror image isomers of a chiral substrate. The selectivity in both shape and intermolecular forces creates enzyme specificity, meaning the enzyme can only catalyze this one specific reaction. These active sites are where substrates undergo chemical reactions by breaking and making new chemical bonds.
After the substrates are picked up by the enzyme can lower the activation energy of a reaction through several different mechanisms. Some enzymes quicken chemical reactions by simply bringing two substrates together in the correct orientation, while others reduce the activation energy of a reaction by bending the substrate molecules in a way that facilitates bond breaking.
Some enzymes can create a favorable environment inside the active site that is favorable to the reaction. For instance, an environment that is nonpolar or one that is slightly acidic. Lastly, some enzymes lower activation energies by playing a role in the chemical reaction themselves. Some active site residues might form temporary covalent bonds with the substrate molecules as part of the reaction.
Once the enzyme and substrates have bound together, they form what is called an enzyme-substrate complex.
The enzyme then releases the product or products of the reaction and is ready to receive a new pair of substrates to start the process all over again.
Here we have the maltase enzyme that is used to make beer. Maltase reacts with the disaccharide maltose to break its alpha-1,4 glycosidic bond to form two glucose molecules.
Once the starch in barley has been broken down into maltose, maltase is required to further convert this disaccharide into fermentable sugars, ultimately allowing yeast to ferment the products of barley into beer.
We hope you enjoyed our video.
And we also hope that you have a better understanding of enzyme specificity.